morning everyone. Welcome to all fourth e-learning classes. This is zoology subject, chapter 4, session 3. We are discussing the good riddance and homeostasis. That is, the process of maintenance of the body fluids and salts in the body is named to be the homeostasis. Good riddance, that is removal of nitrogenous waste. We have discussed that the food that is taken is get digested. Digested food is absorbed. Absorbed food reach the tissues where it become integral part of it and it gets oxidized. And the undigested one are what eliminated outside through the anus. And furtherly, the food substances that are taken in need to be eliminated outside by means of the urine, which is what considered to be the urination, that is excretion. Whatever the food which is reabsorbed is all the nutrients that are reabsorbed is eliminated, sent out of the body through urine. That is what excretion. We have discussed that the proteins, carbohydrates and the fats are taken. In this connection, the carbohydrates and the fats are only stored, whereas proteins are not stored, so they need to be sent out. As they do not store, they need to be sent out. That's the reason why deamination only takes place, not the remaining food. So, why animals needed this? Is the question raised. Generally, in order to carry out metabolic activities, balance of the fluids and the salts is the important phenomenon. For that, animal needs to send out the excess of the fluids and salts. And even this is dependent on the outer environment as well. For suppose, if we see the case with the lower animals, through the body wall by diffusion, the nitrogenous wastes are being eliminated. We have already seen the case with the invertebrates called the protozoans. Amoeba, paramecium. Amoeba and paramecium are eliminated the nitrogenous waste in the form of the ammonia through body wall by diffusion. Then sponges through body wall by diffusion. And the case with the ctenophores and cnidarians, cnidaria followed by ctenophores also through body wall by diffusion. But the case with the platyhelminthes, protonephridia, they are the flame cells which have what is called the flame, the ciliated tubules are present, ciliated tubules, which vibrate like a flame. That's what it is, got the name flame cells, which are also considered to be the protonephridia with the solenocytes. And the case with the nematohelminthes, H-shaped canals, serenity glands. In the case of the annelida, nephridia or the excretory organs. In the case of the arthropods, Malpighian tubules eliminate the nitrogenous waste to the outside, and the coxal glands in the case of the arachnids, as well as the antennary glands or the excretory organs in the case of the crustaceans. Next, molluscans. Remind you one thing, in the case of the mollusca, the excretory organs are none other than the renal glands or metanephridia, pericardial glands it is said to be. And there are examples named to be the organs of Vajanus, organ of Vajanus is also the excretory organs in the case of the mollusca. Organs of Vajanus. And there are certain members in which the Kever organs, Kever glands are also what the excretory organs of the mollusca. Next, in the case of echinodermata, in the case of the echinodermata, the excretory organs are the dermal branchia as well as the tube feet. Tube feet are which are associated with the umbilical system or water vascular system is for excretion. Next, in the case of hemichordates, hemichordates are the members in which the proboscis glands are the excretory organs. Proboscis glands are what the excretory organs. And after that, the urochordates. So far we have seen these are non-chordates. Non-chordates are right from the peripherence to hemichordata. However, protozoans comes under the protista. They are not the kingdom Animalia. However, we have seen. Next, apart from that, till up to here, this is the point to remember. Organs of Bosonus is an excretory organ and Keber glands are the excretory organs. The example we see this in the Unio, Keber's gland, Unio. And echinodermata with the tube feet. Umbilical system or water vascular system is the distinctive feature in the case of the echinoderms, where the tube feet are associated with it and or what play a role in the excretion. Hemichordates are with the proboscis glands. This is the summary of the non-chordates. Now we will see chordates. Chordates are urochordata, cephalochordata and vertebrata. Urochordata, cephalochordata and vertebrata. In the case of urochordates, the excretory organs are what said to be the neural glands. And the case with the cephalochordata are the members in which protonephridia with the solenocytes. It is the point to remember. Next, chordata divided into urochordata, cephalochordata, vertebrata and vertebrata. Urochordata, cephalochordata, vertebrata are the three subphylums. Vertebrata again divided into Anatha, jawless and Gnathostomata, jawed vertebrates. Anatha again Astracodermy and Cyclostomata. Cyclostomates are with the kidneys as the mesonephric kidneys are the excretory organs. And the case with Gnathostomata include fishes, amphibians, reptiles, aves and mammals. Fishes are mesonephric. Mesonephric kidneys. Next, amphibians. Amphibians are with also the mesonephric kidneys. Next, reptiles, aves and the mammals. All these three are with metanephric kidneys. Metanephric kidneys is what we do discuss in this connection. So, the case with the excretory system summary. The case of the all non-chordates and the chordates. We have seen what are the excretory organs. These are what important, which is outside of the textbook information. Organs of Bosonus and Keber glands are to be seen. And also in the case of the urochordates, neural glands. Hemichordates are the proboscis glands. Echinodermata, they are not the organs. Here the example I have given as the part that is associated with the umbilical system is the tube feet, and the dermal branchia are also present as a part in the case of the echinoderms. So tube feet are what locomotory structures which are associated with the umbilical system, helps in the excretion. The case with the cephalochordate protonephridia with the solenocytes, and the vertebrate species mesonephric, amphibians are mesonephric, and the reptiles, apes, and the mammals are metanephric tubules. So the point to remember, the excretory products: ammonia 1, urea, uric acid, guanine, amino acids. Besides that, we do also see, I'll write the list of the excretory substances. Ammonia, highly toxic. Next, urea, less toxic. Uric acid, least toxic. Then guanine is with the spider, guanotalism, and the amino acids, aminotelica, is what the excretory product. So besides that, we do also find certain excretory products. That includes our creatine and creatinine. Next, the ornithuric acid, and apart from that, the other is these are all what we considered to be the excretory product. In this, the ammonia, urea, and uric acid are the major. The remaining or guanine and amino acids are minor, whereas the creatinine, ornithuric acid, and also the bile pigments, bilirubin, biliverdin, these are what also considered to be the excretory products. And the point to remember here, the ammonia, urea, uric acid are nitrogenous waste. We have discussed that the nitrogen is not directly being eliminated. That's the reason why the proteins, amino acids are break down into, that is a deamination of the amino acids. What form? The nitrogenous waste, urea, uric acid. Besides that, the case with the nucleic acids are what undergo the metabolism. What are nucleic acid includes? We are familiar with the nitrogenous bases present in the nucleic acid, purine and pyrimidine. The metabolism of purine and pyrimidine also what forms? The nitrogenous waste. And the case with human, in mammals we discussed that purine metabolism, uric acid is synthesized, purine metabolism. So the case with human beings, by the amino acid deamination, the nitrogenous waste as well as purine metabolism also results in the nitrogenous waste called uric acid. And the other creatinine is considered to be the end product in the muscle contraction. Ornithuric acid, it is excreted by the birds. Next, bilirubin, biliverdin, we know very well that is the worn out RBCs or old RBCs are destroyed in the liver and produces the bilirubin, biliverdin. So this is how these excretory products are sent out and maintains the osmoregulation balance of the salts in the water. This is the summary of this, of the excretory products. Of all these, ammonia is highly toxic, more soluble in water, This is the reason why with more water it is in a diluted form sent out mostly by aquatic animals. We have seen amoeba, hydra, liver fluke, tinea solium, and the bony fishes are ammonia. In the case of the bony fishes, ammonia is sent out in the form of ammonium ions. This is mostly done by diffusion through the gills and the body surface. Least amount is sent out through the mesonephric kidneys. Urea is synthesized by ornithine cycle, which is involved as cyclical reactions in the liver. That is involving the amino acids ornithine. It is one of the cycle of the vertebrate liver. So synthesized, so is it is the hepatic vein contains the blood vessel with large amount of urea is none other than the hepatic veins. Urea is less toxic, that is 1 lakh time less toxic when compared with the ammonia, and it is less soluble in water. And terrestrial habitat has made the animals to lead ureotelism and uricotelism. It is the habitat necessitated them to lead this excretory products. Uric acid is insoluble in water, least toxic, and it is one of water conservation mechanism. And guanine by spider, ureoteric or adult amphibians as well as the mammals, uric acid, tracheate arthropods, land snails, next the reptiles, birds and insects, guanine by spider, amino acids example we have discussed that the molluscans, next the case with the even the allantoin is also one of excretory products we have seen, allantoin. Among the mammals except the apes and the man, they present an enzyme named to be the uricase enzyme which convert the uric acid into allantoin. Then ornithuric acid, orni refers to birds. In that way you can consider it is the resultant of the metabolism of the fats, lipids, and bilirubin, biliverdin. This is what all the examples of the animals excreting their product and maintaining the osmoregulation as well as the homeostasis. Now we'll see the other excretory organs. Other excretory organs in the sense, however, so far we have seen how it is taking place. There are types of the excretory organs, better I say types of the excretory. The list includes are protonephridia, next the mesonephridia, mesonephrica kidneys, and the Malpighian tubules, then the antennary glands, next coxal glands, and the metanephric kidneys, or renal glands is what we see. Okay, so in the evolutionary process, the simple to complex structures are what do see as an evolutionary aspect. We see right from the non-chordates to chordates. See, the early forms are with the exchange of gases through body wall by diffusion. Why? It is a least process of mechanism, since they are microscopic or smaller. As the evolutionary complex takes place, that is, early or multicellular but cellular grade, then tissue grade, then organ grade, then system grade. So as the evolution proceeds, the simple to complex organs have been into the picture of the existence for excretion process. In the case of the protonephridia, these are what said to be the simple. If you see in the case of the invertebrates, here the case with the protonephridia, okay, and Malpighian tubules and antennary glands, coxal glands, if you observe, what you observe is the simple tubular structures are executing excretion. Whereas the case with the metanephric kidney and mesonephric kidney, we find the complex tubular structures are executing excretion. So let me clear you all this one after the another. Protonephridia are considered to be the simple tubular blind structures. These are the what it is to be the simple tubular blind structures. You can see this as a structure resembling in this form. They are what is like a flame. That is the simple tubular structures which are ciliated, present in the body cavity and appear like a flame. So these are what considered to be the flame bulbs. Flame bulbs or flame cells. And these protonephridia are also named to be the with the solenocytes. And these are present in the members named to be platyhelminthes. Example of the protonephridia is platyhelminthes, rotifers, and the other invertebrate members. We can see the protonephridia. Even the case with the lancelets, nothing but cephalochordates. So I repeat, if it is not visible, you just concentrate. What I am saying is protonephridia are simple tubular blind structures, and these are with the tubes which are with the cilia, and these hence are what named to be the flame bulbs. They are selenocytes, seen in the case of platyhelminthes, rotifers, and the cephalochordates. And these are primarily for osmoregulation, maintenance of the balance in the fluids in the body. Mesonephric kidney. Here the point to remember, as the evolutionary progresses, simple to complex is formed. In this connection, mesonephric kidneys are seen in the case of the earthworm, nephridia. Here the point to remember, they are nephridia, typical nephridia, seen in the case of the earthworm. And even the larval molluscans, we do find such kind of the nephridia. And Malpighian tubules, if you observe, in fact, in earthworm, what we considered is earthworm and the frogs are dual excreting animals. The animals which excrete more than two type of the excretory products. The case with the earthworm, when it is in a dried conditions, it excrete the urea. In moist conditions, it excrete ammonia. Frog larvally amniotic, adult it is the ureotelic. That's what dual excreting. And nephridia is a structure which is with nephrostome, body of nephridium and nephrediopore. In earthworm, the typical nephridia referred to as this is the diagrammatic view of the excretory organ, typical nephridia in the case of the earthworm. In the case of earthworm, which is with the ciliated funnel-like. This is a ciliated funnel-like nephrostome. And this is the body of nephridium. And this is a nephrediopore. Nephrediopore is what the typical nephridia is all about, which is seen in the case of the earthworm. Earthworm in the sense there are various species under this. That is Megascolex, Mauritii, Ferritima postuma, Enchytraeus albaeus, Actochetus likewise. Examples are there. Why I am repeating this is, if you go for the Ferritima postuma, typical nephridia is absent. But I said it is earthworm. Earthworm is one species where there are many various species also exist. So this is a typical nephridium structure which is an excreted organ in the case of the earthworm. So it is dual excreting. Next is Malpighian tubules. Let me clear you one diagram related to the Malpighian tubule. This is the alimentary canal. We are all familiar with the alimentary canal in the case of the cockroach. Look at here. This is the midgut, and this is the hindgut. Look at here. So in this connection, these are what said to be the Malpighian tubules. These are what said to be the Malpighian tubules. Malpighian tubules. Malpighian tubules. The blind-ended bundles of 6 bundles of yellow colored structures present in the open into the or blind-ended. It is, I said they are present in the body cavity, means they are not connected to they are present in the coelom, but they do not directly open. The blind-ended yellow colored bundle of structures arise in between the junction of the mesenteron and the hindgut are named to be the Malpighian tubules. These Malpighian tubules are both absorptive and secretory. If I draw the diagram of a single tubule, they are what present in the form of the bunches. That's what they are, bundles. And these are what open to the hindgut. And these Malpighian tubules are example in the case of the tracheate arthropods. We do say, that is, the list we see Malpighian tubules is insecta, Chilopoda, and Diplopoda. I repeat, see, you have to be more concentrated what we are spelling. So certain times if it is not visible, you can hear. Insecta. Insecta includes the insects. That is cockroach and the Anopheles, Culex, all are what insect. Then chilopod in the sense are the centipedes, underleggeds. Diplopods are none other than the millipedes. These are all terrestrial tracheate members are with the Malpighian tubules as an excretory organs, as well as the respiratory organs are tracheal system. That's what I referred as tracheate arthropods, the animal that respire by trachea. So insecta, hylopoda, diplopoda, example Malpighian tubules. Not to confuse with the Malpighian corpuscle or Malpighian body. This is the some funny points certain times which make us to mislead. If I say renin, it's definitely require the spelling. R-E-N-I-N I can write, R-E-N-N-I-N I can write. Both are not same. Renin, Tinidia, Malpighian tubule, Malpighian corpuscle, stapes. Until and unless there is a spelling, biology is of that importance. So be careful. So Malpighian tubule different from Malpighian corpuscle, Malpighian body. Got my point? So that is what Malpighian tubule, insecta, hylopoda, diplopoda. Definitely we'll discuss all this. Already we discussed this renin and renin. One that is related with the digestive process and another with the excretion. Next stapes is also you know very well the smallest bone in the human body is nothing but the stapes. If I don't write the spelling, definitely you may be misleaded. Why means in cockroach also chordate put together podite. That is the mouth part. Be careful. So Malpighian tubules. Let's come to the antennary glands. Antennary glands. That is at the base of the antennae. What is antennae? Antennae nothing but a sensory structure. In the case of the arthropods, antennae, the multi-segmented scape, pedicel and multi-segmented flagella containing antennae. At its base, the gland present named to be the antennary gland, which is an excretory organs in the case of the crustacean. I repeat, crustaceans. Next, coxal glands or excretory organs in the case of arachnida. Arachnida is members related to arthropoda. Arachnida, one of the terrestrial class. Recall your memory. Arthropoda divided into the three subphylums. Trilobata, next Chelicerata, Mandibulata. Chelicerata again, Ziphosaurans and arachnids. So arachnida. Understand? Next, however, this is for osmoregulation. Then metalliferous kidneys is seen in the case of the molluscans and higher group of members called the mammals. So this is the summary of the types of the excretory organs. Protonephridia in the case of the plaque, helminthes, rotifers, cephalochordates, mesonephric kidneys, which are named to be the nephridia, that is earthworm, and the Malpighian tubules. However, the mesonephric kidneys are seen in cyclostomata and fishes and amphibians. So both can be seen. Malpighian tubules, insecta, hylopoda, diplopoda, antennary glands, crustaceans, coxal gland, arachnida, metalliferous kidneys in the case of the mollusca, and the higher members, renal glands, pericardial we refers in the case of the molluscans, whereas in mammals it is metalliferous kidney. It is a evolutionary highest category, metalliferous kidneys, where the excretory organ do not collect the nitrogenous waste from the body cavity.